drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. About who you are and what your lab does?
Yeah, so I'm a professor at the University of Trento, northeast Italy, where I lead a lab which is studying the human microbiome, so all the collection of microbes populating our body. I started to be interested into that more than 15 years ago now, when I joined in the US the Human Microbiome Project. And after that, I've been always more and more fascinated with this topic that we are studying mostly through this approach called metagenomics. And now I lead this lab applying metagenomic approaches mostly on the human microbiome for trying to discover both basic characteristics of the human microbiome, but also aspects that are related with health and disease.
So I know what the Human Genome Project was, but I don't think I've heard of the Human Microbiome Project. What was that?
Yeah, it's actually a bit related, no? Because it was 10 years after the Human Genome Project. At that time, researchers started to appreciate the importance of the human microbiome. So they said, let's sequence whatever is not human inside us, and let's get a map of the human microbiome instead of the human genome. So it started around 10 years after the Human Genome Project. However, it was much more difficult because the researchers realized pretty soon that it was impossible to have a very clear and specific map of the human microbiome, mostly because there is much higher variability in the microbiome than in the genome. So it was difficult to do that. And so the goal of the project changed a little bit toward trying to investigate what is normal to have or not to have in our human microbiome overall.
Yeah, and I suppose that's a huge problem, because it's not simply that there's variability from person to person and that there's many species of microbes, but you've got many different genomes that vary at different locations, even within a single individual.
Yeah, there are really tons of challenges there. There is a lot of variability. Yes, there is a variability between individuals, but also between populations. And there are a lot also of microbes that we never heard about. Microbiologists never cultivated, especially at that time, most of the members of the human microbiome. And so it was difficult to make sense of it. However, what helped a lot was metagenomics, because until the use of sequencing-based approaches, the only way to study the gut microbiome and the human microbiome in general was to try to cultivate the single members. But that is incredibly difficult, and sometimes it's really impossible, because you cannot know, maybe one microbe is interacting with another microbe, so and this interaction is vital for both of them. So you cannot
separate
cultivate only one of them. And so metagenomics, at least from the biotechnological viewpoint, started to solve this issue. So the idea was, let's sequence the genetic content of a sample full of microbes, and by analyzing the genetic content, we can have an idea about which microbes are there. Mm-hmm. But this is difficult, not because, the idea, like with the Human Genome Project, the technology is mostly the same, sequencing the DNA. But sequencing the DNA in the case of the Human Genome Project was only the first step. Then we had to make sense, to reconstruct the full genome, starting from readings of short pieces of the genome. So this is called genome assembly. And is the idea is like a big puzzle, not trying to put together little pieces to reconstruct the full genome. But with the microbiome, is much, much more difficult because it's like trying to solve many, many puzzles all together, starting from, the pieces of the puzzle all scrambled together. And we need to reconstruct many, many puzzles. And each puzzle is the genome of some of these bacteria. And some of these bacteria are completely unknown. So, trying to reconstruct a puzzle without having the picture of what is the final goal.
I see. So the genome is like a puzzle, like, literally a puzzle that you could build with your family on the tabletop. And of course, the pieces are all mixed up, but it comes with a box. So you know what the picture looks like. And I guess with the Human Genome Project, we had a sense for what the human genome looked like. We knew the number of chromosomes. We sort of have the picture on the box. And so that helped in assembling that one genome. But you're talking about metagenomics, which I suppose refers to the fact that, inside of us, there's many different species of microbes in the microbiome. And so you're saying it's akin to having, a bunch of different puzzles all mixed together. And then on top of that, you don't even have a box for many of them because they are brand new species and we have no idea what they're supposed to look like.
Correct. In the gut microbiome, there are usually many hundreds of these different bacteria. And not only bacteria, there are also viruses, fungi, and so on and so forth. So these are the number of different puzzles that we want to reconstruct. And some of the genomes are quite similar. No, it's like the blue sky of many different puzzles. The blue is kind of the same, so it's very difficult even to assign the single fragment of DNA or the single piece of the puzzle to the specific final picture you are trying to reconstruct. So this is to say that basically what kept us busy, our lab and many other labs, was not trying to improve the biotechnological approach of the sequencing, but more analyzing, developing ways to analyze this data. And so we move from being able to know which phyla, so which large taxonomic categories were there, to arrive to specific species or even specific strains or even specific variants. So, distinguishing the nucleotides that are making different, that are differentiating the Escherichia coli in my gut and in your gut, for example.
So it sounds like when you're doing this metagenomic work, the data acquisition side is sort of solved. You have the technology, you can do the sequencing. The problem and the challenge and potentially the interesting thing for someone like you as a computational biologist is really on the genome assembly and data analysis side.
That is the point. That's the reason why I'm a computer scientist and I can apply computer science approaches into this field. It's not only about assembly, because assembly is probably the easiest approach we can think about, but there are many other tasks that we can achieve that are different from assembly. But yes, that is exactly the point. On the technology, the only thing that changed a lot is the cost for sequencing. 10 years ago, it was about 1,000 euros to sequence a sample, a metagenomic sample. Now it's like 10 times less expensive. And this means that we can have much, much larger data sets and codes sequenced, which is at the basis of the big findings, because again, the microbiome is hugely variable. So to make sense of it, you really need to have large sample size.
Yeah, and it's not only variable in the ways that we've mentioned so far, but it's dynamic in a way that the genome isn't. So even though the gene, like, of course, our gene expression can change, genes can turn on and turn off within an individual, but despite all of the expression differences, there's still one genome in all of my cells that's the same genome. But that's probably not the case with the microbiome. The species are probably coming and going, shrinking and expanding to different degrees depending on lifestyle and a bunch of other factors we can talk about. So how do you start to I don't know, how do you start to like standardize things in the lab so that that you're kind of looking at the same thing? Like, do you have to make sure and be very careful about taking samples from the same person in the same state? How do you sort of handle the dynamic nature of the microbiome?
Yeah, that's a fantastic question. So, yeah, in the lab we need to be careful for, for many reasons. So, for example, contamination. Microbes are everywhere, in the air, in the kits, on the bench that you have in the lab. Even if you are trying to start to stay as sterile as possible. So microbes are everywhere, and if you don't pay attention, they will end up in your experiment. Um, on the variability and longitudinal variability of the microbiome, we did a lot of studies, and I think we appreciate right now that the single components in your gut, in your oral cavity, are pretty stable in time in terms of which microbes are there. However, they change a lot in terms of abundances of single microbes, and and the question that we started to be extremely interested in is how do we acquire such microbes now? So if you sequence my gut microbiome, if you sequence your gut microbiome, I'm sure we will have around 20 or 30% of species in common. It's just a little bit if you think about that, no? Because it's like less than half of the species in my gut are also in your gut. But even more astonishing is if you look at the genome of these microbes that we have in common. So I have Escherichia coli in my gut, you have Escherichia coli in your gut. But if we look at the genome, they are only 95% or so similar on the shared genome. So many genes my E. coli has it your doesn't have, and vice versa. At the point that we really think that each human microbiome is unique if we look at the genetic of the human microbiome.
So yeah, so we'll have some species in common, but the majority of them won't be in common. And even for the species that we both have, we both have E. coli in our gut. Basically, what you're saying is they can be different strains of E. coli. So even though they're the same species, they're not really identical.
For E. coli, for example, there are strains that are probiotic, strains that are pathogenic, strains that are commensal, strains that are carcinogenic even, or environmental. And so, knowing which strains we have is important, and that's what actually we are trying to study. And the next step is to understand how do we acquire these strains from. So the baby in utero is sterile, and so instead, a baby of few days of life is already full of microbes. And so we started to focus there to understand what is the process of colonization for our gut and human microbiome in general. And that is what we started several years ago by looking we went into the hospital, and we sequenced the microbiome in many different locations of mothers before they were giving birth, and then the baby just after birth. And we really saw that basically the strains that were present in the mother were those present also in the baby. So with a vertical transmission from mothers to infants that were occurring during delivery. And so, this is to answer your question on what's the variability and longitudinal variability. We start setting a first, kernel of microbes at birth from the mother usually, and then we continuously change it in time.
And so, when the baby is still in the womb prior to birth, you're saying they don't have a microbiome; it's sterile. Do you mean inside the baby, like the gut microbiome, or even is the entire womb devoid of microbes?
It's entirely womb. There are exceptions. Some cases there are some potential pathogens, but in healthy pregnancies, they are sterile. Now, sterile or almost sterile is a small difference.
Very, very, very low microbe.
Very low. So there is not a working microbiome in the baby before birth.
And so is it literally like a physical process? Like the baby is born, it's going through the birth canal, and it's touching the mother's tissue as it's rubbing against it as it's being born, and that's literally what seeds the microbiome.
That's exactly how it happens. And in fact, we see that when there is the C-section instead of natural birth, this seeding of the microbiome from the mother to the baby is not occurring. It's not a very, very big deal because it can still happen in the days after delivery by direct contact. But yes, this is a confirmation that the seeding starts already at birth with the vaginal microbiome, but also with the gut microbiome, which is in any case contaminating many body sites. And we do see this happening. And again, the confirmation is that in the baby, we see the microbes coming, the same exact genetics of the microbes in the corresponding mother, which are different from the strains we see in any other mother. So it has to be at that level of transmission.
Mm-hmm. And so after that microbiome is seeded and it sort of takes root and establishes itself and becomes stable after birth, I mean, I know that the microbiome is plastic. It can change, but how difficult is it under normal natural circumstances for that microbiome to change? So I guess one way of asking that would be, so if a baby's born, it gets its microbiome. If you look 6, 12, 18 months later, 5 years later, whatever, is it more or less going to be the same microbes with the same composition as right after it was born? Or is it going to be completely different? Or is it somewhere right in the middle?
Well, it depends at the age, you know. So infants at birth, they change very rapidly the microbiome. Then they go through changes like breastfeeding or starting to eat food and so on and so forth. So in the first 1.5 years, the microbiome is changing a lot, very rapidly. Then it changes less rapidly, and in the adult is relatively stable at the point that usually, if I look at my microbiome now and my microbiome in a year from now, if I didn't do any big changes in diet or didn't have any medical condition or change content or whatever, we expect to see 80% of the strains to be the same. But there are multiple things you can do to change your microbiome because you want to do that or because it happens. So diet is one big way to change your microbiome. Lifestyle, drugs, of course. For many, think about antibiotics, for example. And then you can arrive to medical ways to change the microbiome, including, for example, fecal microbiome transplantation, which seems to be quite a dirty thing, which is actually what it says. So taking the gut microbiome from an healthy individual, giving it to a diseased individual, and this is a way to reprogramming the gut microbiome quite efficiently, actually.
Yeah. So the microbiome is plastic. It changes quite a bit early in life, naturally. Just, there's a lot of change happening early in life, so it's pretty intuitive. But it stabilizes by the time you're an adult, such that, unless you make major changes to your diet, to your lifestyle, you start taking antibiotics or other things, it will be relatively stable from day to day, from week to week. In a stable, healthy person with a consistent diet and lifestyle, an adult, I'll let you define what exactly this means, but the more important or more consequential microbes, if you were to look at the 1 or 2 or 3 biggest strains in terms of how much they're influencing the physiology of the person in terms of their population density or whatever is relevant here, is there any sort of general principle in terms of like what they're doing? For example, are they primarily preventing pathogens from getting into the body by sort of taking up that space in the gut, or are they primarily digesting critical food components for us? Is there any pattern there, or could it be sort of any of the above, depending on the specific species?
All of these all together. So an healthy, diverse, and stable microbiome is doing a lot of functions, preventing infection, as you mentioned, but also helping us digesting food, producing some compounds, short-chain fatty acids, or some vitamins that they will not be available to us in any other way. But also, priming and setting the immune system is an extremely important function that has implication even on tumors, for example, because an immune system in a positive and healthy configuration can prevent diseases from autoimmune diseases, but also to cancer and much more. and then there is the gut-brain axis, for example. So the interaction between our gut and our in our head by this gut-brain axis. So, the function of the human microbiome are very, very big. Metabolism much more, and an healthy microbiome does all these functions all together. Also, though some that I am sure we still have to discover completely.
Mm-hmm. So given how prevalent antibiotics have been for many years now in our society, it's pretty well known that this is obviously affecting the microbiome. It's not just affecting the pathogens that we intend to use the antibiotics against. But if you take broad spectrum antibiotics, you're basically, clearing out your microbiome or greatly reducing it or affecting it in some way. When that happens, given that it's happened to many, almost everyone has taken an antibiotic at some point in their life.
Yeah.
But when that happens, so let's say I take an antibiotic tomorrow, a high dose of a general antibiotic, and then I get better. I'm no longer sick, and then I recover. How likely is it that my gut microbiome is going to recover and be the same basic population it was before, or is it very likely to be completely different?
Yeah. So antibiotics are clearly a huge problem, especially for inducing pathogen resistance. So some pathogens become resistant to antibiotics because of the use of antibiotics themselves. But instead, in terms of the microbiome, yes, you kind of reset the microbiome, and then the microbiome, after you end taking antibiotics, starts to rebuild. I wouldn't say from scratch, but from the seedings from the seeds that remain there.
Mm-hmm.
And I would say that most of the time, which we can give percentages here, probably between 70 and 90% of the times, you go back to an equilibrium of your gut microbiome, which is similar to what you had before.
Mm-hmm.
But the problem is that 10, 20, 30% of the cases in which you reach another equilibrium can still be a good equilibrium, maybe in some rare cases even better than the original ones.
Mm-hmm.
But in other cases, it's not. And so this may induce allergies or other problems that are due to that. However, I don't think actually, antibiotics are the drugs with the worse impact on the gut microbiome, because
Really?
antibiotics, you take them for a couple of weeks maybe, but then you stop them. There are other drugs that are, you assume them, for years or even forever. For example, proton pump inhibitors, they are changing the pH of your stomach, and they are allowing many different microbes arriving to your gut. And it is something that is not like, again, 2 weeks like antibiotics, but is for many, many months. And this is impacting much more the microbiome. So we can find from the microbiome, we can predict whether you are under proton pump inhibitors at very, very high accuracies. So there is a signature of it.
And I would imagine something like a proton pump inhibitor, it's changing pH. That's a very broad spectrum stimulus. It's going to basically affect any microbe species that would be there no matter what.
Correct. And also changing the acquisition of microbes, no? Because the way we change our microbiome, coming back to the previous topic, how is our microbiome changing? So if you think our microbiome, the members of our gut microbiomes are not living in the environment. It is not that, on this table, or if I go into the woods, the microbes that are there are also the microbes in my gut. 99% of the microbes in our gut can only be in the gut of other individuals, or maybe other primates, but we are not in touch with primates, no? So basically, a study that we did recently really showed that is the person-to-person interaction leading to the exchange of microbes and the changing of our microbiome. So we discussed before, mother transmitting microbes to the infant, but even as adults, we interact, and as we very well know, that we exchange pathogens, from viruses, SARS-CoV-2, whatever. We exchange also the other microbes.
Yeah.
And we know, in a household, when a pathogen arrives, SARS-CoV-2, for example, 99% of the cases it gets to the whole family.
From one person to the next,
it's the same with our gut microbes. And that is the way we are seeing. So, for example, if you take two individuals like you and me that we never interacted directly, we probably don't have even one strain in common. But if we start to live together into a house for a couple of years, statistically, we'll arrive to share 20% of our gut microbiome, even just because we share the same physical space.
I see. So a couple things pop up for me there. So it sort of makes intuitive sense that if you share a common space, a common environment with other people, your microbiomes will converge to some extent. So there's a sort of a two-pronged question there. To some extent, that could just be shared environment. We're both eating the oranges on the table, the same foods in the fridge. We're naturally going to converge on diet to some extent. So naturally, that will facilitate a convergence to our microbiome to some extent. There's also the question that you brought up of direct transmission. Can I catch my gut microbiome from you literally? And I want to ask you about that. So again, if we think about other pathogens, respiratory viruses, things like that, very simple, very intuitive. If someone has a respiratory illness, they sneeze or they cough, I'm going to inhale those viral particles or whatever, and it goes from their lungs to my lungs. It's a little bit more difficult for me to think about how this happens with something like the gut microbiome, because as you said, many of these species are adapted to live in my intestine. They probably can't, I would imagine, they can't live on my microphone. They can't live on my hand. They can't even live in my mouth if they're adapted to, say, my colon or my small intestine. How does one transmit their gut microbiome if that's the case?
Yeah, so that is a fantastic question. So we know very little about that because we just discovered recently that there is this huge transmission of the microbiome in general. But first of all, the oral microbiome is more transmissible than the gut microbiome. So I said that if we stay together for 2-3 years, we share 15-20% of our gut microbiome, but we are probably going to share 40-50% of our oral microbiome.
I see.
Well, this actually measured when is a partner that are also kissing, but even in any case, sharing the same space will lead to higher sharing of the oral compared to the gut microbiome. But if you think still of the pathogens, even bad gut bugs are transmitted almost in the same way, with the same efficiency that the oral pathogens or the respiratory pathogens. So, if you go back even many years, microbiologists developed this theory that all microbes are everywhere, and then the environment selects. This can be a specific application of this theory. So, because we put our microbes everywhere because we just need maybe few spores or few cells of gut microbes that are in the environment, and this will be enough to have the microbe, if it arrives into your gut, to find the perfect environment to actually grow. So when we study transmission, we are actually unable at the moment to disentangle what is the effect of my microbes arriving to your gut and the probability that transmission will actually result in a long-standing colonization. And without being able to disentangle these two effects, it's going to be very difficult to understand what are the main sources and modalities of transmission.
Interesting. Yeah, it makes sense. I mean, obviously, if the baby picks up the microbiome from the mother through direct contact, I mean, people who live together have lots of direct contact together, especially, if you imagine your spouse or your partner, you have direct mouth-to-mouth contact, and that's going to influence the composition of the microbiome.
But you have to, the microbes transmitting between adults, they need to break what is called colonization resistance, because each of us, you have your microbiome, which is well established. It has an equilibrium. It's not so easy for someone to arrive and to kick one strain out and colonize there. Yeah. The baby is favored by the fact that it's not colonized by anything, so it's much more likely that whatever arrives there will actually colonize. And this is the same with antibiotic resistance. So we saw that babies at the daycare, after they take antibiotics, they are much more prone to acquire microbes from their peers, because basically they are resetting the microbiome, they are lowering the colonization resistance. So the other incoming strains are much more likely to colonize.
Wow. And to the extent that the microbiome that's transmitted to you can cause you to develop or lose a given phenotype or trait or health outcome, it seems plausible that in human society today, the dual use of drugs like antibiotics or like other drugs, like you mentioned, proton pump inhibitors, plus the interactions we have with different individuals, you could essentially catch things from other people if you become prone to acquiring the microbiome, if you just went through an antibiotic treatment or some other drug treatment, for example. And I guess maybe one way we can sort of start to talk about this is, I know that there's work out there, for example, showing that in rodents at least, you can basically transmit obesity from one mouse to another. If you give the obese mouse's microbiome to the lean mouse, and the lean mouse becomes obese. And those types of experiments have been done for obesity and other things, as far as I'm aware. To the extent that's true in humans, that means, in theory, right, you could have a nice, healthy, lean baby, say, or child. They get sick, you give them antibiotics, you sort of clear out or diminish their microbiome, and then they go to daycare. And if they're around someone, another child who's obese, they could catch their obesity. Is that possible? To what extent do we think something like that is actually happening?
Yeah, we need to be a bit careful here. But for sure, what we know is that our social interaction and our lifestyle is impacting our microbiome by really, the people we interact with, because we are catching the microbes from there. And yes, there are several diseases that are linked with the microbiome. However, if it is not a single pathogen, there are very, very few diseases that are only due to the microbiome.
Okay.
So it is a complex system. But yes, for some conditions that are causally linked, at least in part, with the microbiome, then yes, microbiome transmission makes these diseases that are usually called non-communicable diseases at least partially communicable. And that is actually what we are studying right now, because as I was telling you, we started discovering this microbiome transmission, and now also other scientists added exactly your same idea. Maybe part of these non-transmissible diseases are actually transmissible. We are studying this. We are studying it from cancer to obesity to metabolic diseases.
Yeah, because normally, I mean, this is the definition of a non-communicable disease. You don't normally think about catching cancer from someone or catching metabolic syndrome from someone. But what we're talking about here is the possibility that there's a context-dependent possibility that otherwise non-communicable diseases can become communicable. And the microbiome.
Let's say, I would say that maybe the risk for the disease becomes a bit communicable.
I see.
You mentioned cancer. There are some cancers that are caused by microbes. And so in that cases, it is known. But these microbes are usually not pathogens, are microbes that are enhancing the risk for developing certain kind of cancers. And so it could be that if you live with a person that has these bacteria that are slightly more prone to develop cancer, then maybe you can get them.
And how exactly does that work? Are these microbes producing a metabolite that is mutagenic or something like this?
Well, in the case of cancer, there are many different ways. It can be by inducing inflammation, and inflammation in turn may be a risk factor for cancer. There are some microbes that are known to indeed basically damage the human genome, and that can cause cancer. And there are other bacteria that are thought to be protective against cancer, and so not having them may be an indirect way to enhance the risk for cancer. The microbiome in oncology is a relatively novel topic, and relatively means probably 5 to 10 years. Again, if we exclude the pathogen part, and this is very, very quickly developing. Also, because we have to think about the microbiome as also something to exploit for treatment. I'm referring, for example, to the cancer treatment. The immunotherapy is one of the latest fantastic discoveries in the field of oncology. Immunotherapy is saving life of people right now. So people with melanoma, for example, advanced melanoma, that are not responding to any other chemotherapy or other therapies, some of them, a good fraction of them, are responding to immunotherapy, completely cleaning out the cancer. But in some cases, this is not working. And we saw that the microbiome, in some ways, is linked with responding or not to immunotherapy. And this is extremely important. And to prove that, some very recent trials did fecal microbiota transplantation from an healthy donor to someone who was not responding to immunotherapy, and after reprogramming the microbiome via FMT, fecal microbiota transplantation, some of these individuals started to respond to immunotherapy.
So the immunotherapy is not working, and then you change their microbiome, and then the immunotherapy is now effective, and it wasn't before.
Correct. So this is linked with what you were saying before. No? So FMT means basically inducing a transmission, is artificial transmission. Is not by being taught in contact with someone, is really, artificial transmission. And this induced some changes in the microbiome and in turn in the immune system, which made immunotherapy starting to work. And this is proven and is more and more an important topic in oncology.
Do we know exactly how that works? Is the new microbiome stimulating the immune system to operate in some new fashion? Is it producing a molecule that wasn't there before?
Yeah, the answer is that we don't know, unfortunately. And that's the reason why we do FMT, which is transmitting everything. Because if we knew which are the features and the important thing, we would just transplant that, or basically having some new probiotic kind of intervention targeting directly that. So clearly, of course, there is a lot of research here because we want to understand. But my feeling is that this is not going to be very successful because our human microbiome, as again and again, is unique to each of us. So
full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.